Good afternoon, everyone. It is our privilege and absolute pleasure to welcome Professor Kim Nasmyth, our keynote speaker for today. He will be introduced in more detail by Kaylin Chong, who is the president of the Oxford Biological Society.
Cool. So Professor Kim Nasmyth is a fellow of the Royal Society, and he was also an Oxford University Whitley Professor of Biochemistry and head of the department from 2006 to 2011. Through his work on gene expression, as well as his discovery of the anaphase promoting complex, Professor Nasmyth's research has provided valuable insight into the healthy cell division processes, as well as key proteins. In addition, his discovery of cohesin, a protein complex responsible for mitotic and meiotic chromosomal segregation, has helped shape our understanding of how sister chromosomes are held together until segregation occurs in daughter cells. As an exceptional contributor to the advancement and understanding of molecular biology, especially with regards to chromosome segregation, we are honored and delighted to give students and the interested public a chance to hear from you at this inaugural joint Oxbridge Symposium. So please welcome Professor Nasmyth.
Okay, shall I start?
Yep.
Okay, and thank you very much for that nice introduction. Somehow, yeah, okay, good. I'm not quite sure where, oh, that's good, right. Get rid of these pictures. Okay, well, welcome to you all, and my sympathies for you being in this situation, all of us being in this situation. I've done one of these talks before, and I think I've watched others of myself, and my experience of it, that is, it's slightly easier for the audience than it is for the speaker, because the speaker has no idea whether the audience has fallen asleep or not. And that's the slightly tricky thing. I'll do my best to prevent that. Well, I just want to start off with the big question. Last year, I think, was the 75th anniversary of Schrodinger's What is Life, a very famous book that inspired Francis Crick to go into biology, where Schrödinger asked the question is, what was it that made life special? He concluded that the main thing was the distinguished life from non-life was its ability to maintain homeostasis and thereby defy the second law of thermodynamics, requiring living organisms to feed on negative entropy. I don't know how many of you have read Schrödinger's book, but I would recommend it to anybody who hasn't. But there is, I think, one feature of it that is sort of somewhat missing, is that he underemphasizes the crucial thing that distinguishes living systems is not so much their defiance of the laws of thermodynamics, but their ability to reproduce. Now, this is obviously poignant at the moment because are viruses alive or are they not? And obviously viruses don't maintain homeostasis in the same way that living organisms do, but they do reproduce. So really both of these things are key features, and viruses have one and living organisms have, actual cells have both. Of course, evolution is simply a corollary of reproduction. I think it was Karl Popper who pointed out that actually there is no doubt, there can be no doubt about evolution. It is a logical consequence of the fact of reproduction. I should be able to change, oh, there we go. So one of the obviously once it was realized that all living organisms were made of cells, and that cells didn't arise spontaneously, but new cells arose solely by the growth and division of pre-existing cells. This was one of the most exciting things that happened in the 19th century. It's the basis of all modern medicine, and is the foundation really of all modern biology. And one of the most striking features when people started studying the process of cell division was the observation that Whereas when a cell divides, essentially the cell gets bigger and then it sort of splits into two, roughly in 2 equal halves, but it doesn't have to be precise. And basically half of the cell goes to one side and half goes to the other side, and the two daughter cells end up with material that was in the mother cell. But when you actually looked inside cells, if you had a good enough microscope, you noticed that within the cell was the nucleus, and the nucleus did not divide in the same way. And as Walter Fleming described in 1879, what's remarkable of the nucleus, it didn't just get bigger and bigger and then divide into two. It actually underwent this extraordinary morphogenesis, which we now describe as mitosis. And this was beautifully described by Walter Fleming in 1879, the transformation of the nuclear material into these threads, the realization that these threads were paired side by side, and that the two pairs of each thread, called Faden in German, we now call them chromosomes, but they're originally called threads, that the two sister threads, as it were, moved to opposite poles of the cell before it divided. Now, this is beautifully described in Fleming's paper in 1879, but what is lacking in Fleming's paper is any sort of question saying, well, why do cells do this remarkable thing? Why do the contents of the nucleus undergo this extraordinary choreography, this extraordinary morphogenesis? And it wasn't until a few years later, in probably one of the most important theoretical papers in the entire history of biology — there aren't very many important theoretical papers in biology — Wilhelm Roux addressed what was the meaning of this, what was referred to as indirect nuclear division. It was indirect because it involved this transformation into threads. And he asked the question, a rather naive question, why have such a complex process if simple division would do the job? And it was Roux who first really groped his way towards the notion that this process of mitosis might have something to do not with the quantitative division of the cell, but rather the qualitative division. Namely, he suggested the mitotic figures are the mechanisms which allow the nucleus to be divided not only quantitatively, which of course, in the case of the cell, you get so many thousand ribosomes in one half, another so many thousand ribosomes in the other half, but according to the mass and nature of individual qualities. And you can see in his picture here how these individual qualities, he's suggesting that this precise division was about dividing up individual qualities. Now, one shouldn't read too much into this paper. You won't find the modern theory of chromosome theory in the paper, but nevertheless, it is really the beginnings of thinking seriously about heredity. I'm sure most of you have never heard of this paper, but it's very, very short, and I recommend you go and read it at some stage. Now, I would say the next key step was made by Theodore Boveri, and it was Boveri really who I think you could describe as the discoverer of the genome. It was realized that all of our cells contain a characteristic number of chromosomes. And Boveri realized that these numbers were constant. And there was a great debate about, and people thought, well, the chromosomes may, how many chromosomes do you need for a cell to divide? And there was, because there were actually great variations in the number of chromosomes that cells had. For example, an Indian muntjac has, I believe, 7 chromosomes, and a Chinese muntjac has 22. And yet they both look like muntjacs. And so the question was, why do some cells have, or some organisms have fewer chromosomes than others? Maybe you just need 1 chromosome and the rest are all just sort of backups. And it was Boveri who, by studying systematic chromosome missegregation in the early zygotic divisions in the sea urchin, proved essentially that the sea urchin development depended on each cell acquiring at least one haploid set of the chromosomes. And it was Boveri who really showed that you need a complete set of chromosomes. You don't need a diploid set, but a haploid set was necessary, and in the case of the sea urchin Pluteus, sufficient for development. And I think that can be really, this discovery took place in 1902, which, as many of you know, was when Mendel's laws were rediscovered. And it was Boveri and Sutton, an American scientist, who both pointed out the parallel between chromosomes, the segregation of chromosomes, and Mendel's determinants. And that was really the beginnings, the birth of the chromosome theory of heredity. Now, probably the next crucial step didn't take place for several decades. And this was, of course, Avery's demonstration that DNA was the substance responsible for pneumococcal transformation. This was in 1944. Again, roughly the same time that Schrödinger wrote his book. And so then this was the point when people suddenly realized that it was the DNA of chromosomes that may be the crucial ingredient. And it was really then just a matter of time before the structure of DNA was elucidated by Watson and Crick. Schrödinger, in his book, What is Life, sort of worked out that the gene must be very small, and therefore there was a very small number of atoms involved in it. And he said this posed an enormous problem. If you have very small numbers of atoms, and if you read your Boltzmann, how on earth can how can such small number of molecules confer such stable order? And this was a conundrum, which was the heart of Schrödinger's book. And he postulated at the time that therefore, that when we understood the laws of heredity, they might require a revision of the laws of chemistry and physics. And one of the interesting things, of course, is the truth was much more prosaic. DNA was just a chemical, and we didn't require any revision of the laws of chemistry and physics. And Schrödinger himself was extremely disappointed in this as a physicist. And when Crick wrote to him saying, look what we've discovered, he never replied. I think he was so disappointed, the fact the truth was so prosaic. Now, so that's really going from chromosomes to DNA. It's a story most of you are familiar with. And I really want to talk about another thing, and this is we go back to this process of cell proliferation. And as you all know, during the proliferation of the cells, each DNA molecule has to be replicated, and the two sister DNAs must be then segregated to opposite poles. And this has to take place together with, and has to be coordinated with the growth of the cell. But there are two fundamental processes for the propagation of the genetic material. One is replication, the other is segregation. Now, the double helix provided obviously a theoretical framework to understand the replication problem, namely semi-conservative replication. It's turned out that the enzymology of replication is much more complicated than we ever anticipated, and this is still a very active area of research, but the fundamental principle emerged from the structure of DNA. However, at the process of chromosome segregation, there was no insight gained from the structure of DNA as to how those sister DNAs would be moved to opposite sides of the cell during mitosis. And the segregation problem has turned out to be a much more difficult problem to understand. Now, one of the reasons why it's turned out to be such a difficult problem is that that, for example, in 1958, this would be 5 years after the double helix, even then it was inconceivable that each one of our chromatids in a chromosome would contain a single DNA molecule. I show here a picture of a model that basically people thought that the chromosome would be made up of small segments of DNA that would be then attached by some other chemical linker, be it protein or whatever. Nobody could conceive that a molecule could be so large that it would be a single DNA molecule in a single chromatid. Nobody could conceive how you could solve the unwinding problem that had been pointed out by Delbruck. Namely, how do you unwind the DNA in order to replicate it? And Delbruck had pointed out this problem soon after the double helix was published. And it is a very deep problem. We now know how the problem is solved. We know about topoisomerase and so forth. But in 1958, nobody knew about that. So this was really the model of chromosomal DNA in 1958. And it wasn't until the early 1960s when people started to look at DNA spilling out of bacterial cells, first of all by electron microscopy by Kleinschmidt, and secondly by autoradiography by John Cairns. You've probably seen this picture, but not this one, but this is really the more important one. Essentially, you could see the DNA spilling out of a single bacterial cell, and as far as Kleinschmidt could tell, there were no ends, and it all appeared to be a single molecule. And it was therefore in the early 60s that people realized that chromosomal DNAs were much larger than they had ever anticipated. And subsequently, it was demonstrated, of course, that each one of our chromosomes, of which, of course, there's even more DNA than a bacterial one, is composed of a single DNA molecule. Now, the fact that there's such an enormous amount of DNA in one of our chromosomes causes one to realize the nature of the problem of segregation problem. And to appreciate this better, I just want to draw an analogy. I'm currently at the moment in the south of France, where I have a second job making wine. And vines are rather unruly plants that need to be kept organized by a trellising system where you have wires on a series of posts. Now, when, of course, you replace your vines, you've got to pull out the wires and replace the vines and the wires as well. And you take out a few hundred meters of wire and you end up with this tangled mess. Now, I want you to consider, just imagine this wire was a molecule of DNA. Now, the diameter of DNA is 2 nanometers. And the diameter of this wire is, say, 2 millimeters. And I think most of you probably know that there are 5 centimeters. The average chromosome in a human is about 5 centimeters long. That's 5 centimeters of DNA. And so if that DNA was a wire like this, it would be 50 kilometers long. That's for a human chromosome. 50 kilometers long, 50 kilometers, this is just this is maybe 100 or 200 meters. Can you imagine the mess and the tangles that would be associated with a 50 kilometer long wire? And that's for humans, but the poor Mexican axolotl, whose chromosomes are 20 times larger, it would be the equivalent of 1,000 kilometers of wire. And yet the cell can deal with chromosomes, be they DNA, whether they're 50 kilometers long or 100 kilometers long. So this is one of the fundamental problems, is how is the DNA organized such that it doesn't get all tangled up? And how do you separate the sisters from each other? And so here we now know, of course, that the chromosomal DNAs occupy specific territories. Even in interphase, they're not a sort of just tangled wire, they're highly organized even in interphase. But then when the cells undergo mitosis, the 2 sister DNAs end up being assembled into cylindrical chromatids that are attached along their sides. So you have a pair of sister chromatids, as had been observed by Walter Fleming in 1879. So what is the basis of the chromosome metamorphosis? Not only going from tangled DNA to a chromosome territory and from chromosome territory to a mitotic chromosome. Well, there are a number of problems that one needs to consider, and I'll just go through these very quickly. One problem is if you're just going to sort of, people refer to this process as chromosome condensation. But if it were mere chromosome condensation, the DNA would end up being spherical or a ball, but it isn't. Chromosomes are cylindrical. So why are they cylindrical and not spherical? A second question is that you're going to pull on these DNAs via kineticals attached to microtubules. And in particular, in meiosis, you're going to pull along the longitudinal axis of those chromosomes, which are called bivalent chromosomes. And the DNA doesn't just unravel like a ball of wool would when a kitten plays with it. So something holds this whole thing together and generates a certain longitudinal stiffness or elasticity. What confers that elasticity? The third problem is how on earth does the DNA at this end of the chromatid know it's on the same molecule as the DNA on this end? That is, how are all the DNAs from a single molecule, one sister, packaged into one chromatid, whereas the DNAs of its sister molecule packaged into a separate one? How is that achieved? This is referred to as chromatid individualization. Lastly, the process of mitosis requires that once those chromatids have been packaged, the sister DNAs have been packaged into chromatids, they are nevertheless held together by sister chromatid cohesion, which then enables the mitotic spindle to pull them in opposite directions in mitosis. So for example, if you don't have cohesion and the two chromatids were separated, there's no way the microtubules will be able to pull them in opposite directions. And as you all probably know, the logic of mitosis is because you have sister chromatid cohesion, and if you pull the sister kineticals in opposite directions, you generate the tension, which will stabilize the connection between kineticals and microtubules. And if you don't pull them in opposite directions, you don't create that tension, and so those connections are unstable. And that's why sister chromatid cohesion, by resisting spindle forces when they pull in opposite directions, which is the correct way to do so, that's how sister chromatid cohesion enables mitosis to take place. But the key point is that most segregation of sister DNAs takes place long before microtubules come into play. And that very same process is what's going on in a bacterial cell. Somehow, all of this DNA, which is one sister DNA, has largely been segregated away from its sister, even in the absence of microtubules. How do you generate these mitotic structures? Now, you're all familiar that the DNA is packaged into nucleosomes, and you know about the structure of the nucleosome. And I'm sure all of you were brought up, certainly when you were at high school, and probably also at the beginning of your university career, on what has been the model that's been in the textbook for probably 3 decades. Namely, that the chromosome was explained in terms of the structure of the nucleosome. Well, I have to say this model is complete fantasy. There was never any evidence for it. And what's more, it never explained anything. The model never actually explained how it was. The properties of nucleosomes didn't explain how you magically went from here to here to here. It was never a model, but it's there in the textbooks. It's referred to as a model, but actually it was a complete fantasy. And I'll come back to why I think this lasted for so long. Well, one of the reasons why we know this is completely wrong is from beautiful work from Tatsuya Hirano, who could assemble mitotic chromatids in xenopus extracts. And by various tricks and means, he could deplete the nucleosome assembly apparatus from those extracts. And even though the DNA was no longer packaged into nucleosomes, as in when you deplete this particular nucleosome remodeling protein, the DNAs were still assembled into chromatid-like structures. Now, of course, bacteria don't have nucleosomes, and they also have to organize their DNAs and can segregate sister DNAs. So it never made any sense that nucleosomes would explain anything, because bacteria don't have nucleosomes. So how then are chromosomes organized? And I think the first clue came again within, I think it was Walter Fleming, again, who noticed this. Within a year or two of describing mitosis, he described meiotic chromosomes, in particular a period of meiosis called diplotin, which is a region time in meiosis when you've undergone recombination, you've generated bivalent chromosomes, and then these are in an oocyte, and they're undergoing very high rates of transcription. And so there's an enormous amount of RNA and proteins associated with the genes when they're transcribed. And as a consequence, you can actually see in the light microscope, even in living tissue, these amphibian lampbrush chromosomes. And you could see they have an axis around which there are a series of loops. And these are so-called lampbrush chromosomes from diplotin in amphibians. So this then led, by one means or another, to the notion that actually not just lampbrush chromosomes, but all mitotic chromosomes are built up by a series of loops that are assembled round an axis. And Ulrich Lemley in 1977 proposed there was a proteinaceous scaffold around which these loops were arranged. And so that was the fundamental structure of the mitotic chromosome, not this in C, but this in D, namely a series of loops associated with a scaffold. And I'm sure this model has also been in the textbooks for a long time, and I'm sure you have been it has been mentioned to you in your lectures or at school. Now the trouble is, and indeed, if you extract nucleosomes from mitotic chromosomes, you can see loops, and you can see an insoluble material that might be Lemley's scaffold. The problem is that there were two fundamental problems with the scaffold hypothesis. One was an empirical problem, namely the scaffold hypothesis proposed that there was a proteinaceous axis around which the loops of DNA were arranged. And then John Marco, as a physicist, he isolated newt chromosomes, which are very large, and held them together on the end of little needles and pulled on them and could measure their elasticity. And then he added nucleases and cut the DNA down to a certain level. And if the scaffold model was correct, then basically the longitudinal axis of the chromosome shouldn't unravel. You'd just break up the DNA, but the DNA would still be associated with the proteinaceous scaffold. But what he discovered was that DNA cleavage down to about 10 to 15 kilobases completely destroyed the structure of the chromosome. And that was completely contrary to the scaffold hypothesis. An even deeper problem with the scaffold hypothesis is, even if it did exist, how would all the DNAs from a single molecule be attached to the same scaffold? In other words, the hypothesis had no explanation for how DNA is actually organized. And in short, it never provided an adequate explanation for how chromosomes were built. Well, at this point, I want to quote to you a very famous French scientist who died a few years ago, Francis Jacob. You're probably familiar with his work on the lac operon and lambda phage. A very, very, very fine scientist. He wrote a wonderful book about his life in science called The Statue Within, which I'd recommend any of you to read. And in there, I was reading this a few years ago, and I found this this sentence, which suddenly meant a lot to me. I hadn't realized, and he put it very well. He said, "The process of experimental science does not consist in explaining the unknown by the known, as in certain mathematical proofs. It aims instead to give an account of what is observed by the properties of what is imagined." Now, some of you may think this is a lot of sort of French philosophy and a lot of sort of mystical hocus pocus, but actually it's extremely deep. What Jacob is saying is our task as scientists is to realize that the world you're looking at cannot be explained in terms of what we already know. That is the task of a true scientist, is to realize you cannot explain the world in terms of what you know. That's what Newton did. He said the movement of the planets could not be explained by concentric circles and all that hocus pocus. And he then said, but I'm going to imagine an alien, imaginary, mystical force called gravity. I'm going to give it these properties. And if this force existed, I can explain the movement of the planets. And that was the beginning of modern science. It is this leap of imagination to postulate a new force. And this is what Jacob is saying, and this is the heart of science. And lest you think that I've completely lost my mind, I was reading an article in The Guardian this week. Some of you may have already seen it. There was a big excitement about some chemical they found on Venus that might or might not be a sign of life. And there was an article by Charles Cockrell, who was the professor of astrobiology at the University of Edinburgh. Anyway, hidden in this article is the sentence, science demands that we build hypotheses based on existing knowledge and not construct in our minds aliens to fill the gaps. Well, can you see that actually Cockrell is saying exactly the opposite of Jacob? And I would say that Cockrell just doesn't understand what science is about. And what I would also like to say is that this example of the chromosome being built out of nucleosomes was precisely an attempt of science to build a hypothesis based on existing knowledge, and a failure to recognize that existing knowledge could not explain what you wish to explain. So this was a prime example of the above statement being completely wrong. Okay, that's so much for philosophy. Well, enzymes that one and the new forces that one had to invoke to induce this transformation turned out to be a new set of enzymes, and the enzyme that does it in mitosis is this enzyme called condensin, which you probably learnt about. It's a very different beast. This is a nucleosome to the same scale here, and this is some of the subunits of condensin associated with DNA here. And you can see it's a very different beast at all, a very totally different type of molecule. So how does condensin, which has this sort of shape, how does it mediate this extraordinary transformation? And this was largely the work of Tatsuya Hirano. Well, you may have heard of the idea now that condensin, as well as its cousin cohesin, about which we'll talk more later, they both have this ability. Well, the idea was that both of them had this ability to create little loops of DNA and then extrude those loops until they got larger and larger. This was actually a crazy idea that I had when I got back from a climbing trip in the Dolomites many years ago, and I had the temerity to write it up in a review. And it's now referred to as loop extrusion, namely that condensin assembles all that DNA into a mitotic chromatid through this very simple activity, namely it builds a little loop, and then it's a DNA translocase and somehow creates that loop, causes that loop to get ever larger until all of the, and neighboring condensins, they don't speak to each other particularly, they just do their thing, they extrude loops until you go from this situation to this situation here. And essentially, we now know from theoretical work from Leonard Nerney and John Marco and other physicists that this idea of loop extrusion can indeed fully explain how it is that condensin turns that mass of tangled mass of DNA into chromatids. More recently, people have been able to demonstrate that condensin can extrude loops on individual molecules of DNA. These are single molecules attached to glass slides. You add condensin, and then when you have a buffer flow, what normally you will see is condensin, if it extrudes loops, it will tighten that DNA up. And so you go to this structure here. And if while you do that, you then flow, you flow buffer in, you can see that this DNA here is actually a loop that's been formed, and you can see it being extruded. And so we now know that condensin does have this ability to extrude loops in vitro. And it appears that it will extrude DNA within the loop very rapidly, up to 1,500 base pairs per second. Now, how it does it is still totally unclear, and I'll have a little bit more to say about that in a moment. I think most of you have probably heard about the notion that condensin and cohesin are loop extruders, so I won't dwell on this any further. So it turns out, so it is condensin that is building these structures, probably by loop extrusion in mitosis. But those DNAs, chromosome DNAs, need to be organized in interphase as well. They're not just a tangled mass of DNA, nucleosomal DNA. They must be organized into chromatid-like structures, even in interphase. And whereas condensin is a loop extruder in mitosis and generates mitotic chromosomes, it's cohesin that organizes DNAs during interphase. And condensin and cohesin turn out to be related complexes, and we'll say a little bit more about their relative structures in a moment. And for example, if you can take a cell in interphase where we've known for a long time that the chromosomes are organized in territories, but it turns out all you have to do is to delete a regulatory subunit of cohesin that causes cohesin causes its processivity of loop extrusion to be reduced, because if you have this subunit, the cohesin will do loop extrusion and then it will drop off the chromosomes and start again. But if you knock out WOPL, cohesin goes on extruding loops and never drops off the chromosomes, and the cohesin, in the absence of WOPL, will actually turn the interphase DNA into chromatids. Here's the DNA, and here the cohesin accumulating along the longitudinal axes of those chromatids. So cohesin basically does exactly the same as condensin. It's just doing it in interphase, and because WOPL causes it to drop off, it's not completely processive. It doesn't actually cause the DNA to be turned into chromatids. But if you knock out WOPL, it will. This interphase cell remains an interphase cell and life goes on. So chromosome condensation per se does not stop the cell from growing. Okay, so, and also more recently, cohesin has also been demonstrated to be a loop extruder in single molecule experiments in vitro. And this has all been published quite recently. So again, I won't dwell on that. Now, condensin and cohesin are first cousins. They all belong to the SMC Klysin family of proteins. Bacteria have members of these families, but the ones, cohesin and condensin are sort of first cousins within this family. And both of them are composed of heterodimers of SMC proteins, SMC2 and 4 in the case of condensin, a dimerization domain up here, which is called the hinge domain because you form this V-shaped SMC2,4 heterodimer, and this is referred to as the hinge. It's a bit of a misnomer. It's basically a dimerization domain. Likewise, cohesin is a heterodimer between SMC1 and 3. Now, the extraordinary thing about these SMCs in the case of condensin and cohesin is that the ATPase, that's the apices of this is long coil. These are intramolecular coils, and the N and C termini of these molecules come together to form one half of an ABC-like ATPase. So you have an ATPase down here, another one down here. And the ATPases at the apices of this V are interconnected by a so-called Klysin subunit. It's called baron in condensin, it's called SSC1 in cohesin. As a consequence, both of them form a tripartite ring structure composed of a V-shaped heterodimer of SMCs with the Klysin connecting the apices of the ATPase at the end of those Vs. Now, in addition to that, both complexes are regulated by related proteins, which are large hook-shaped proteins that are composed of heat-repeat regulatory subunits. They're called hawks, because they are heat-repeat proteins associated with Klysins. Hawks, heat-repeat proteins associated with Klysins. And in the case of condensin, there are 2. In the case of cohesin, there are 3. In the case of cohesin, there are only ever 2 at any one time. It turns out that SSE2 or NIPBL in humans, or PDS5, these are mutually exclusive. So you either have SSE2 here or you have PDS5 here. So in both cases, you only have 2 hawks associated with the complex. And cohesin and condensin are special and different from the SMC5-6 complex in eukaryotes and from bacterial SMC complexes, in that they use these hawk proteins to regulate the complex, and they don't use a different set of proteins that the bacterial and the SMC5-6 complexes. So this is why the condensin and cohesin are really very closely related in evolutionary. Well, it has emerged that the coils of those associated with the SMC proteins of both condensin and cohesin are actually not straight coils. There are breaks, and in particular, there is a so-called elbow region around which the coil is folded. And in fact, the coil is folded around this elbow. And this is true both for condensin. This is cryo-EM work from Jan Lerver's lab at LMB, and this is for cohesin. The elbow is at a slightly different place for the 2, but in both cases you have a clearly defined elbow, and the coil is folded back. And that's true for both cohesin and condensin. It's also true for the bacterial, the E. coli protein muck beef, which is a sort of related protein in E. coli. So it's a very conserved feature of these complexes. The other feature of these complexes, and so here we have the hinge, we have these coils, which generally spend a lot of their time zipped up together. You have an elbow around which you have folding, and here you have the ATPases here, the chlysin associated with those, and then these hawk regulatory subunits that are recruited to the complex by binding the chlysin, but then they regulate the ATPase down here. Now, whether the zipping up of the coils and the folding of the coils is something that is regulated is not known, but they are clearly important features of the complex. Now, the other feature that is very, very important to realize is the ATPase heads at the ends of those coils can be in 2 states. They can either be disengaged, namely when there is no ATP present. Here is the head of SMC1, the ATPase of SSE1. This is the N-terminus of the chlysin associated with the coil as it emerges from the ATPase. Here's the C-terminus of the chlysin, and the rest of the chlysin interconnects like this to create the ring structure. Here is the SMC1 ATPase, and this is in the disengaged state. But in the presence of ATP, but only under certain particular conditions, and in the case of cohesin, you need both SSE2, the hawk SSE2 or NIPBL, and DNA and ATP. Then the ATP bound to one of the heads, the phosphates bind signature motifs in the other head, and vice versa. So the 2 heads will engage in the presence of a pair of ATP molecules. The 2 ATP molecules, as it were, bring the 2 heads together, and you create this so-called engaged state. Okay, and so there are these 2 different states, disengaged and engaged. And this is very, very important, and it will be fundamental to how these complexes can, as it were, bind and disengage DNA in an ATP-dependent fashion, and thereby be DNA translocases. I hope that's clear to you. Now, I want to talk about, I don't want to talk too much about loop extrusion because we really have very little idea of how it works. And I now want to turn to the mechanism by which cohesin holds sister chromatids together. So cohesin, like condensin, is a loop extruder, but it was discovered for its function, which is to hold sister chromatids together. It turns out cohesin has these 2 very different activities, and it appears that when cohesin is loop extruding, it cannot hold sisters together, and vice versa. They seem to be 2 mutually exclusive states of the complex. And so I want to talk about how cohesin holds sister chromatids together. Now, and this is the process, this is the importance of cohesin. It's cohesin holding those sister kinetochores together, enabling the the spindle, the microtubules, to attach to opposite poles that enables this wonderful process of mitosis. And the crucial point is the key event in mitosis is the first step is the alignment, which depends on this tug of war between microtubules and sister chromatid cohesion. And then the next step, which is the metaphase to anaphase transition, is triggered by the activation by the anaphase promoting complex of this enzyme, this protease called separase. And what separase does is it just clips the cohesin of cohesin and opens up this ring structure. That's all separase does, and that's what destroys sister chromatid cohesion. Now, what follows will all concern experiments that we've been doing in the budding yeast, Saccharomyces cerevisiae, but we believe everything inside a yeast cell is pretty similar to what's inside a human cell. It's just easier to study. And many years ago, when we realized that cohesin formed this ring structure and that separase, by being activated by the APC when all the chromosomes have aligned, what it does is it simply clips the cohesin subunit in two. We proposed that cohesin might hold sisters together by entrapping sister DNAs inside the ring. And we call this the SMC cohesin ring, or the SK ring. And that is the ring model. We've spent many years trying to test this hypothesis. And the best experiment we've been able to do has been the following, is to introduce at these three interfaces that create the tripartite SK ring, introduce cysteines. We had to get, this was all done in collaboration with Jan Lerver's lab. We got crystal structures of these three interfaces. Thereby, we were able to design cysteines, which we could introduce into those interfaces, show that they were functional, introduce those proteins into yeast, and then we're able to use a bifunctional thiol-specific cross-linking reagent that goes into cells, into living cells, and will cross-link these rings inside the cell. Once you've done the cross-linking, of course, the cell is dead because it cross-links all sorts of other cysteines together, but it actually will cross-link it in a living cell. Now, the ring model suggested that if you were to cross-link these three interfaces and then pull down the cohesin, and you had a yeast cell in which you had a small circular mini chromosome, which generated sister chromatid cohesion, and if the DNAs were trapped inside that ring according to the ring model, and if you had chemically circularized the ring by cross-linking these interfaces, you could take these IPs and denature all the proteins, but the cohesin, which had been circularized chemically, would be denatured. The nucleosomes would be denatured, they'd fall off, but the DNAs would remain catenated by this chemically circularized ring. And these DNAs, even though they had been decatenated by topo 2 inside the cell, would run in an agarose gel as a dimer, plus a bit of scrambled egg associated with this circularized cohesin. And so we were able to do this. We introduced these cysteines here in this interface, these ones here and here. Cells were viable. We could show you could get about 20% of the molecules would be fully circularized by treating with this cross-linking reagent BMOE. And then when you take a cell which contained cysteines, 6 cysteines, we call this the 6C experiment, cysteine here, 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 and here. If and only you had all 6 cysteines, now you got two different, this was the monomeric supercoiled DNA in the IP, which would run as a supercoiled DNA. Even though the cells had undergone replication, topo 2 had done its business and decatenated the DNAs. They decatenated the DNAs with regard to DNA-DNA catenation, so all the DNAs would run as monomeric DNAs. But if you'd chemically circularized the cohesin, now you've got a new form, which we called catenated dimers. And the DNAs ran as dimers by virtue of them being entrapped inside this cohesin ring that had been circularized. In addition, we got a more modest shift in the electrophoretic mobility caused by the catenation of a monomeric DNA by a single cohesin ring, and we call these catenated monomers. And it's these that we're going to be seeing quite a lot of in the next few slides. So remember what these are, these catenated dimers. A single DNA molecule has been catenated by a cohesin ring, which you can then chemically circularize and show that even in the presence of SDS, that circularized cohesin ring will delay the electrophoretic mobility. So the question is, several years ago, James Collier in my lab, who was a graduate student, asked, could he get cohesin to trap DNA inside its ring in vitro? And so what he did is, and this was again in collaboration with Jan Lerver's lab, we made the cohesin trimers in baculovirus. We then made SSC2 or NIPBL in baculovirus. We made SSC3 or the SA proteins in humans, also in baculovirus. And we added the trimer plus SSC2 and SSC3. And we added circular DNA, and we added ATP, and we asked, could we get DNAs trapped inside those rings? Well, we made a 6C version of it so that we could, with BMOE, we could chemically circularize the cohesin ring, and then we would treat with BMOE, we denature the proteins and run the DNAs on the gel. And if you had 6 cysteines there, if you didn't have the 6 cysteines there, then all the DNA ran as monomeric DNAs. But if you had the ring and these regulatory subunits plus ATP, if you added BMOE and if you had 6C there, then now you got CMs, that is, this was the naked DNA, there was very little of that left. Now you've got a ladder of retarded bands. And this was the CM, this was a single DNA trapped by a single cohesin ring. This was 2 cohesin rings on a single DNA, 3 cohesin rings, and so forth. I hope that's clear to you. A very simple assay, but this absolutely proved that you were looking at entrapment of DNAs inside this cohesin ring. And you needed to have all 6 cysteines. If you had the 5C control, it didn't work. You needed to have ATP. If you didn't have ATP, it didn't work either. It depended on both SSC2 and SSC3, both of which are required for entrapment in vivo. And here's entrapment in the absence of SSC2. Here's entrapment in the presence of both proteins. Here's entrapment in the presence of just SSC3. There's very little entrapment compared to here. And likewise, here's what happens if you don't have SSC3. There's very little entrapment compared to the control here. Now, so therefore, James was able to reproduce in vitro entrapment of DNAs inside these rings in a manner that depended on all the things you required for entrapment in vivo, namely ATP hydrolysis, SSC2, and SSC3. And we believe that this entrapment process is fundamental to building cystochromatic cohesion. By some means, after replication, you end up with sister DNAs trapped in a single ring, and I'm going to say a little bit at the end of this talk about how that might happen. But in the meanwhile, want to consider something else. And that is, though you need entrapment of DNAs inside the SK ring to build cohesion, because we believe cohesion is co-entrapment of sister DNAs, it turns out you can make mutant forms of this complex which are no longer able to entrap DNAs inside the ring. But these mutant forms are defective in cystochromatic cohesion, but they are not defective in loading cohesin onto chromosomes in yeast or for loop extrusion in vitro, as has been recently showed by Jan Michael Peters' lab in Vienna. So there are clearly ways for cohesin to associate with chromosomes, and in particular to produce loop extrusion that does not involve entrapment of DNA inside the SMC-Kleisen ring. So are there other ways that DNAs can be associated with the cohesin ring? And so what James did was the following. And here we need to now consider what his original assay had cysteines here, 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 and here, and he was looking entrapment inside the SK ring. But it turns out, as I told you, that those heads can be engaged in the presence of ATP. And if those heads were engaged, and if the hinge was dimerized, you would create a subcompartment in that ring, which we call the ES compartment for engaged heads. And this is the SMC compartment. So if you had cysteines here that would be specific for the engaged state, and you had cysteines here that were specific for the hinge. If you combine those two sets of cysteines and you cross-link here and here, then the question is, could DNAs be trapped in the ES compartment? Likewise, you could have cysteines that were specific for engaged heads, and you had cysteines that were specific for the cohesin associated with this head and the cohesin associated with this head. And you could ask, could DNAs be trapped in this compartment, which would be called the E-cohesin compartment? So you can divide the SK ring, if the heads are engaged, into an ES compartment and an EK compartment. And so what James asked was, could he see entrapment? He'd shown there was entrapment in SK rings. Could he see entrapment in ES or EK compartments by just putting cysteines in different places? And what he found was that the cohesin would trap DNAs in both of these two ES and EK compartments, and that this entrapment was 20 times or more rapid than the entrapment in SK rings. In SK rings, it takes about 40 minutes to get lots of entrapment. Here's 2 minutes, here's 40 minutes. If you do exactly the same reaction, but instead look for ES entrapment or EK entrapment, you can see the reaction is complete within two minutes. So ES and EK entrapment can also be measured and takes place much more rapidly than SK entrapment. Now, the ES and EK entrapment, it turns out, is completely Scc3 independent. So Scc3 is essential for entrapment in the SK ring, but for ES entrapment, minus plus Scc3, minus plus Scc3 EK, no effect whatsoever. The entrapment in the ES or the EK compartment is completely independent of Scc3. But that entrapment, even in the absence of Scc3, is strictly dependent on Scc2. ES in the absence or presence of Scc2, EK in the absence or presence of Scc2. So basically, DNAs get trapped inside either the ES or the EK compartment, and all you need is ATP, the trimer, and Scc2. And this is a very rapid reaction, and it does not require Scc3. Interestingly also, that unlike SK entrapment, the entrapment of DNAs in the ES and EK compartments does not require ATP hydrolysis. You can make these EQ mutations whereby the ATP combine, but it can't be hydrolyzed. So the heads get engaged and can't disengage. And SK entrapment is greatly reduced by these mutations, but both ES and EK entrapment is completely unaltered by making these mutations that prevent ATP hydrolysis. You need ATP because you need ATP to bring the two heads together, but you do not need ATP hydrolysis. And so it turns out that entrapment within the ES and the EK compartments, which is driven by Scc2 alone in the presence of ATP, in the absence of ATP hydrolysis, is not accompanied by entrapment within SK rings. So if you do the same reaction, you take the trimer, and you just have Scc2 and ATP, and you can get ES and EK entrapment, you will get absolutely no SK entrapment. So here's the conundrum. How can you get entrapment of DNA in this compartment and this compartment at the same time as you do not get entrapment in this compartment? You may be a little confused. I know most people who I try to explain this to are confused by this. But it turns out there's an incredibly simple explanation. What if you take a tripartite ring and the heads are disengaged, the cohesin is folded up, and now the DNA is passed upwards from the south to the north and passed through the heads, after which the heads engaged, the DNA sits on top of those heads, and now you have engaged heads. If you cross-link here, you cross-link here, the DNA will be trapped in the ES compartment. If you cross-link here, you cross-link here, and you cross-link here, the DNA will be trapped in the EK compartment. But you will note that when the DNA enters the ES and EK compartment, I have not opened any of the interfaces of the SK ring. Therefore, the DNA cannot be entrapped inside the SK ring. And that's how we explain how you can get ES and EK entrapment without SK entrapment. Well, you may think this is all very rather topological and difficult to understand, but actually what's going on, turns out, is now being revealed by cryo-EM structures from Hung Tao Yu's lab and also from Jan Lerver's lab in Cambridge. And in particular, Jan's lab has solved the structure of the cohesin complex, or the heads, the region around the heads, by cryo-EM. In this particular case, you've got heads that can't hydrolyze ATP. It's a DNA oligonucleotide. You've got SSE2 there, but this is essentially, and you've got the cohesin there, it's essentially the reaction we do for ES-EK entrapment. That is, it's the cohesin trimer plus SSE2 and ATP, but no ATP hydrolysis. And what you can see is that when the DNA is trapped in the ES-EK compartment, what has happened is the DNA is indeed has been clamped on top of the engaged heads, and it's been clamped there by SSE2, one part of which binds to the SMC1 head, and the other binds to the coil of the SMC3 here. And here's the sort of molecular model. But this is a 3.4 angstrom model. This was just published in eLife. And it turns out that they were also able to see we'll also get lower resolution cryo-EM structures of DNA clamped onto these rings in the presence of SSE2, even with a circular DNA. You see the circular DNA, and you see these cohesin complexes clamped onto that circular DNA. There are half a dozen of them on this circular DNA. They got a lower resolution structure of the clamped state, even on circular DNA. And you can see the DNA here in class averages, that you can see the DNA is not bent. You've simply got a circular DNA passing through the ring here. And here's the DNA clamped on top of the heads and clamped there by SSE2. Well, it turns out this clamped state, you can also see, again at lower resolution, that the coils of the ring have been folded back, and it turns out that the hinge contacts the SSE2. But the significance of this, we don't yet know fully. But the key point is, in this clamp state, the coil is folded back. So really what we're saying is that there is this SSE2, the activity of SSE2, or NIPBL as it is in humans, is to basically bring the DNA between these two heads and clamp them on top of the heads to form that structure you saw in those beautiful cryo-EM structures. And what we suggest is that, clearly this clamping is something that will take place in the presence of ATP, but upon ATP hydrolysis, the DNA would be released from the clamp. And clearly, for a DNA translocase, you've got to have an ATP-dependent DNA binding site. And we propose that every time cohesin translocates along DNA, as it must do during loop extrusion, it is drawing DNA into the ES-EK compartments like this, clamping the DNA in there by virtue of SSE2 and the engaged heads. And every cycle of DNA translocation, it involves a clamping, unclamping, and each cycle involves the successive clamping of DNA further along the chromosome. So we believe this is the heart of the motor of the DNA translocase that's involved in DNA translocation during loop extrusion. Well, how on earth does DNA get into how does this process take place? We don't know, but there's a very interesting structure that, again, that Jan's lab has got of the condensin subunit, a condensin with a folded coil. Here are the ATPases, and the equivalent subunit in condensin to SSE2 is this protein called YCG1. And they found a form of the condensin complex in which the heads were pushed apart in the disengaged state, and the YCG1 was, as it were, bridging them, was sort of splitting them apart. Now, YCG1 is the equivalent subunit of SEC2, and if SEC2 were also to push those two heads together and form this bridge state, then as shown here, if SEC2 were to form the bridge state and then forms the clamp state, if you went from the bridge state to the clamp state, and the DNA were to bind to this binding site in SEC2, which we know exists, then you can see how it is that the DNA would be drawn into the clamp state by SEC2. So we believe this may be the heart of the engine whereby the DNA is clamped by those heads. That's just a hypothesis. Now, we believe, therefore, that the first step crucial to loop extrusion is the formation of this clamp state and then the disengagement of DNA. We believe also that the clamp state takes place, is a very rapid step and is involved in loop extrusion. But if SEC3 comes along, then somehow, in a much slower step, sometimes the DNA now enters the cohesin ring. Namely, one of the interfaces of the cohesin ring opens up, and that DNA is passed through one of the interfaces of the ring. Well, how does this happen? We don't know. Here's that clamp state. I've drawn it with a folded coil. And to go from the clamp state, which is produced by SEC2, to produce SK entrapment in the presence of SEC3, somehow you've got to open up an interface of the ring and pass DNA through that interface. It's either the hinge interface here, namely it opens up and you pass the DNA through, and now you've got entrapment in the SK ring, or you have to open up a cohesin interface with the ATPase. And we don't know which it is, but somehow, sometimes this clamp state is associated by passage of DNA inside the ring. And that is fundamental for building sister chromatid cohesion. Now, all this work was done by James Collier, but it was a very close collaboration with Byung-Chul Lee, who did all the cryo-EM in Jan Nerva's lab in Cambridge. And in the last 5 minutes, I just want to say something about how do you build cohesion during replication. And the crucial point is that cohesion is built during S phase. You can express cohesin. If you express cohesin during S phase, then cohesin will trap sister DNAs inside the ring. But if you express cohesin in G2, even if you've previously expressed, even if the sisters are still held together by cohesin that was expressed in S phase, but this cohesin that's expressed in G2 post-replicatively goes onto chromosomes, will trap individual DNAs, but will not trap sister DNAs. So cohesion and co-entrapment of sister DNAs inside the ring, which is what we believe cohesion is something that only takes place during S phase normally. So there's a big question, what happens during S phase? During G1, some of the cohesin molecules have trapped individual DNAs, they form CMs, and then magically after S phase, you form catenated dimers, and namely sister DNAs get trapped inside the same ring. The question, one of the questions we've asked in recent years is how do you produce this state? Is this ring derived from a ring that had trapped an individual DNA before replication, and you converted this structure to this structure during S phase without the ring dissociating from the chromosome? Or is this ring loaded on to the sister DNAs after the fork has passed de novo? That was the question we asked ourselves. And essentially what we've been able to develop an assay in yeast whereby we can actually measure this form, namely catenated monomers, essentially being converted to catenated dimers inside yeast cells. And we've been able to show that this event we call conversion. It's a bit like the passage, the nucleosomes that are ahead of the fork being passed on to the sister DNAs, both the leading and the lagging strand. But unlike the histones, where this is probably not terribly important, this process in cohesin is fundamental to building cohesion. So some of the cohesin that is on the chromosome before you replicate, when the fork comes on, it is converted into cohesin that traps sister DNAs after the fork has gone. That is, this is converted to this. And what's more, this process depends on specific proteins associated with the replosome. So, for example, this conversion process depends on TOF1, CSM3, CTF4, and CHL1. That's these proteins. They're all proteins associated with the replosome. They're required for this conversion process. And the rate-limiting protein of this thing, if you overproduce it, you get more of this kind of conversion, is the CHL1 DDX11. This is DDX11 in humans. It turns out to be a helicase. Now, what's a helicase doing in this conversion process, we don't yet know. But that's a very important clue. But crucially, if you knock out this conversion pathway by making mutations in any one of these genes, the yeast cells are still viable. They have cohesion defects. You don't build normal cohesion. You have defects in cohesion, but you still enough cohesion to live, and something is keeping the cell alive. And it turns out what's keeping the cell alive is there's a second pathway. And the second pathway depends on SSE2 being active. The conversion pathway, you need SSE2 to load cohesin onto the chromosomes in G1, but you don't need SSE2 or the ATPase to convert that chromosomal cohesin to these cohesive structures here. So it's, as it were, SSE2 independent. And that's the conversion pathway. But there's a second pathway which requires other replosome-associated proteins, namely this alternative RFC called the CTF18-containing RFC that's associated polyepsilon part of the replosome. These proteins are required together with SSE2 to load cohesin de novo and to load on and build catenated dimers or co-entrapped cystodNAs by taking cohesin in the nucleoplasm and loading it on at the fork. So essentially there are these two different pathways that build cohesion at S phase. There's the conversion pathway and the de novo pathway. So at the end of the day, cohesion establishment is so important, you have to have two quite separate mechanisms to do it. It is a belt and braces job. You can use a belt to hold up your trousers, or you can use braces, or you can use both. And in English, we say something is a belt and braces job is when you want it to be done so well that even if one system fails, the other system will also work. And this discovery about cohesion establishment being a belt and braces job, and the discovery of this conversion pathway and the de novo pathway and the particular replosome proteins that are involved, was all discovered by Madhu Srinivasan in my lab, and that also has been published in eLife rather recently. So that is everything I wanted to say. I'm sure I went on too long. I have no idea how long I went on. I apologize if I've gone on too long, but I'd love to take some questions.
Thank you, Professor Nasmyth, for your very informative and thought-provoking talk. I'm pretty sure people would be willing to keep on listening, even if you want to go on for another hour.
No, I'm going to shut up now. I'd like to hear what I'd like you to ask me some questions.
All right. So, well, there was one question on YouTube, but the person mentioned, perhaps you have already answered the question, but I'll just read out the question just in case you would like to add anything. So the question is, are the loop extrusion and entrapment functions of cohesin related, or we believe the same molecule evolved to perform two independent functions?
My guess, condensin probably doesn't entrap DNAs inside its ring. And
cohesin, sorry.
Yeah, I know. I'm going to answer the question is the loop extrusion and the DNA entrapment function, are they separate functions? It's a single complex. Cohesin does both. It does cystochromatic cohesion, which needs entrapment in the SK ring. It does loop extrusion, which does not. Now, I think it's important to realize that The bacterial proteins do loop extrusion, condensin does loop extrusion, cohesin does loop extrusion. So I suspect the primordial function of these complexes is loop extrusion, which does not involve entrapment of DNAs inside the ring. And that and therefore cohesin has this primordial function, and that's one of the things cohesin can do. But cohesin acquired a separate function, namely to entrap DNAs, which it uses for sister chromatid cohesion. And therefore, I think that I'm not quite sure how this evolved. I can't tell you how it evolved. I suspect that cohesin actually evolved. My guess is that cohesin evolved before condensin. It evolved from a bacterial precursor that did loop extrusion. It evolved for the purposes of meiosis, where of course it plays an incredibly important role in organizing recombination and for building meiotic chromosomes and regulating the whole recombination process, and of course building sister chromatid cohesion. My suspicion is that cohesin and sister chromatid cohesion and its whole role in loop extrusion, that all evolved in meiosis, and then it was subsequently, condensin is a derivative which lost one of the functions of cohesin and became a pure loop extruder. And then condensin was then used for mitosis. So mitosis came after meiosis in evolution. Most people think meiosis is more complicated than mitosis, and therefore it must have come later. But though we may never know this, I'm sure meiosis came first and mitosis second, because these eukaryotic cells evolved from cells that could segregate their chromosomes perfectly normally, like a bacterial cell. So why would you develop a second system? So it's clear this system must have evolved for some other function, and then it was co-opted for chromosome segregation. And that other thing was, of course, is recombination. And that's what meiosis is all about. So my guess is cohesin really evolved a cohesin for its meiotic functions. And it needed to do both loop extrusion and entrapment, and then you had a gene duplication event, and you produced a version of cohesin that couldn't do entrapment, and that was condensin, and then it only did it in mitosis. That's my guess as to how it evolved, but that's only a guess.
Thank you. So we have one question from YouTube from Amanda. She said, thank you very much for your talk. And the question is, why do you think that misinformation, like the example you gave earlier, makes it into school textbooks?
Well, I think, you see, most scientists don't really understand what science is really about. This guy writing in The Guardian totally misunderstands what science is about. He thinks it's about explaining the world in terms of what we already know. And that is what most scientists think their job is. But if you want to discover something new in life, you've got to get that idea out of your head and realize that if you want to push back the frontiers, it's a question of realizing when you can't explain the world in terms of what you know. But the point is that most scientists includes most people who write textbooks, and they write and they try to explain. And textbooks, of course, are deeply misleading for students, because they try to explain to the student that we understand the world, and they try to build an explanation of the world in terms of what we already know. And that's why those images appear in textbooks, because it is an attempt to explain to students the world in terms of our existing knowledge. And this is why reading textbooks is so terribly bad for you to learn how to become a scientist. There's a lot of knowledge you've got to learn, and textbooks are a shortcut, but don't believe half of what you read there. Does that make sense? I hope.
It does make sense to me. Yeah. So we have two questions at this moment. One, so both from Zoom. One, Mirta said, thank you for a fascinating talk. I have read a little on the subject of TADs, for example, CTCF sites leading to the anchoring of condensin. However, the topic seems to be a little controversial when considering TADs and gene expression. What is your opinion on the subject? Specifically, do you believe that condensins may be so dynamic that the single cell view will be very different to population views of whole genome, in brackets 3D structure?
Well, TADs are the structures that you see when you do high-C. I mean, high-C is this technique where you treat the cell with formaldehyde, and then you cut up the DNA and you ligate the ends together, and you see which DNAs were close to each other. And you can do that for the whole genome, and you find there is regions where sequences are more likely to be co-ligated together than regions that are outside those. And it turns out that TADs are created by cohesin, and they're created by cohesin doing loop extrusion. But the cohesin, the key thing about cohesin, unlike condensin, is there's this protein called CTCF, which acts as a roadblock for the loop extrusion. So when cohesin is extruding a loop and it comes up to a CTCF, and the CTCF is pointing in a particular direction, then the cohesin stops there. It may not forever, but it gets delayed there. And essentially, TADs boundaries are created by CTCF sites that point in opposite directions and ensure that cohesin will loop extrude within this interval, but will not extrude beyond that interval. And that's what a TAD is. I think a TAD is created by sequences being brought together by cohesin in the process of loop extrusion. And the greater level of ligation that you see inside TADs that you see in high-C maps is due to that's happening. They're not real things, because actually what you're looking at is one particular cohesin in a particular place inside that TAD in one cell and another one in a different position. A TAD is not a stable loop that then creates this structure that is stable. Sometimes when the cohesin gets to the ends, it will get to that point. But most of the time, the cohesin will be somewhere in the middle. And that's what generates the TADs. Now, the idea, what is the purpose of CTCF regulating loop extrusion? And the idea is that enhancers can be a long way away from promoters that they activate. And cohesin, by loop extruding, could bring an enhancer close to a promoter a long way away, and it might be the wrong promoter to be brought up against. And so what the cell does is it tries to insulate, it tries to prevent cohesin from bringing distant enhancers up against non-cognate promoters. So for a promoter to be activated by a cohesin, by an enhancer, it has to be in the same TAD, namely, it mustn't be separated by a CTCF site that would block the loop extrusion. Now, it's all very controversial because it turns out that it's very unclear. If you knock out CTCF or you knock out cohesin, essentially there's not major changes in transcription and not major ectopic transcription. There aren't. How important insulation is still very unclear. It's probably important for certain genes at certain stages of development. But nobody really has a really good example of a gene where they can demonstrate it's really, really, really important. Now, the one place I think that we do know loop extrusion as regulated by CTCF is really, really, really important is not in the area of transcription, but is in the immune system. And it turns out that our ability to battle the coronavirus depends on the diversity of antibodies and VDJ recombination. And it turns out the process by which different V genes are recombined to DJ junctions, which is mediated by the recombination, is you've got this fundamental problem is the V genes are megabases away from the recombination site where associated with the D segment. And that's where the recombinase starts out. And so what happens, it turns out, is that the reg recombinase rides along with cohesin, which is extruding loops and goes all the way through until it reaches a V gene. And the problem is it's got to go an awful long way, and sooner or later the cohesin will drop off because WOPL causes it to dissociate. And it turns out that in order for V genes to get recombined with DJ segments, it turns out that the lymphocyte that is involved or the precursor cell, has to downregulate WOPL, this subunit of cohesin, which causes cohesin to drop off the chromosome every now and then. And when it downregulates WOPL, now the loop extrusion can be processive. It can get all the way through to the V genes, and then it can go for one V gene or another V gene or another, and that can be random. So essentially, this process of loop extrusion is absolutely fundamental in the process by which the rab recombinase can bring V genes together with the DJ segments. And so the whole functioning of the immune system would not work without loop extrusion. And here it's absolutely clear, it's absolutely fundamental. And downregulation of WOPL is essential for this so-called locus contraction process. But all locus contraction is basically downregulating WOPL and allowing the loop extrusion to be more processive. So that's my answer to those of you who wonder of the role of loop extrusion. I think the jury's out as far as transcription, but the one area we know it's absolutely fundamental for is in the immune system. So go and read those papers is my advice, and all will be clear.
Thank you. One question from Zoom. Is cohesin involved in liquid-liquid phase separation? I heard some theories to do with chromatin packaging that involve this process and would be very interested to hear your thoughts.
Well, it has been proposed that cohesin and condensin can form condensates. I think most proteins mixed up with other polymers will form condensates. People have known this for a very long time, protein chemists. The question is this a physiological process? And the real question is does cohesin and do condensin form condensates inside cells? Now, in the case of cohesin, people have started looking quite carefully, particularly when it's doing cystochromatic cohesion, as to what are the number of cohesin complexes that are involved when it is holding cystochromatids together. There's a very nice paper from Anne Villeneuve's lab where they looked in meiotic cells in C. elegans. And they concluded that the cohesin complexes that were conferring cystochromatic cohesion in meiosis were individual cohesin complexes, which is completely consistent with the ring model, namely you have a single ring with two DNAs inside it. No cohesin condensates. So I think for cohesion, this is nonsense. It's just an in vitro artifact. And I don't think it really explains anything very much. But time will tell whether it has any function. But I don't believe it has any role in cystochromatic cohesion. It turns out that cohesin is actually involved in loop extrusion by cohesin, probably breaks up condensation of nucleosomes. So there's this tendency of inactive nucleosomes to cluster together and to condensate, and active ones, these so-called AB compartments. And cohesin activity breaks that up. So basically loop extrusion will break up nucleosomal condensation. That may be one of the functions of loop extrusion in interface cells.
Thank you. And so one question from YouTube from Lucas. Are there any examples where the conversion pathway leads to a stalled replication fork and alternative origin firing?
That's a very good question, and I think the answer is we don't know, but I think this is a very active area of research. We would need to alter the cohesion. I mean, normally it doesn't, but there are suggestions in the literature that it does. I'm not entirely convinced by any of them, but I think this is a very interesting area. And in particular, the role of cohesin and the conversion pathway versus the de novo pathway in terms of how double-strand breaks or stalled replication forks are repaired or restarted, I think is going to be a very active area of research in the future. I think what's going on with cohesin at the fork is extremely important for how DNAs get repaired at forks. And this is an area I think will be very active in the future, but very murky and mysterious at the moment.
Thank you. Probably one last question from YouTube. Do you think this DNA replication mechanism only apply to modern organisms, or are there fundamental aspects to it that make it relevant to explaining the first replicating molecules or origin of life?
Well, I mean, I don't think the first living organisms had DNA. I think whether it was an RNA world or we don't know, but it almost certainly wasn't DNA, and therefore, I don't think, I don't think it would have been involved. I mean, DNA replication came along late in the evolution of life, for sure. So I think it's it's a very important aspect of all life as we know it now. But one suspects that the earliest forms of life did not involve DNA replication. But the trouble is, we can all speculate, and people can do their fancy experiments in a test tube. But the problem is what actually happened. This is a historical question. The library in Alexandria burnt down with all the information, all the books inside, and a lot of the intermediate steps in evolution burnt down, as it were, in the course of evolution. So unless we find the missing links, it's going to be very difficult to work out what happened. I think this is the deeply frustrating aspect of studying the origin of life. It's not really a science, it's a metaphysics at present. But if we found life elsewhere that had a very, that's why everyone's so excited in astrobiology. But I don't have too many hopes, I have to say.
Thank you once again. So I guess this is the end of our Q&A session. Thank you very much for the questions, and thank you, Professor Nasmyth, for answering those questions. So I'll pass the floor to Christoph to do the wrap up of this session. Thank you.
So first of all, Professor Nasmyth, thank you so much for that empirical feast, starting off with the 19th century discoveries of threads in nuclei through to recently published papers on the structural details of cohesin and condensin. So, and also including, of course, your own approach to science, to empirical science, and how it requires imagination to take those important steps. So I'd also like to thank everyone who attended this talk with us, and to everyone who came up with questions, to everyone who engaged with our speaker today. And if I can also thank everyone for joining us in our first day of Varsity Sci so far. I hope you've enjoyed it, and I'll probably end the live stream here. Thank you very much, everyone, and hopefully I'll see you at the next session and at the rest of the during the rest of the event.
Thank you very much. It's been a pleasure. It's remarkable what you can do under these circumstances. And my great deepest sympathy to you all that most of your interactions for the next 6 months or even next year or two will be at this level. We're learning how to do it. It's remarkable what you can do. And thank you for inviting me. At some point, I hope we get more intimate, as it were. And good luck this next year, all of you.
Thank you so much.
Thank you.